Okay, so what I want to start with is then how do cells end up with these chromosome breaks? The simplest way in which chromosome breakage could occur is if a DNA replication fork, which starts here and moves down the DNA, encounters one of these nicks, a break in the DNA. Here, simply an interruption on one of the two strands. This is now one DNA molecule, Watson and Crick, and as the replication comes to this point, it cannot progress beyond this nick. And as the replication fork then comes from the other side to this point, what one ends up with is a double-strand break. This kind of interruption is not simply something that happens where a nick is introduced into the DNA by accident, but in fact, there are certain drugs, anti-cancer drugs such as camptothecin, which cause this kind of interruption by blocking the action of the protein topoisomerase 1 and leaving a covalent attachment of the protein to the DNA. And as the replication fork reaches this point, there is the possibility of creating these kinds of double-strand breaks. Stalled replication forks can be produced in a large number of ways. The two that I've illustrated here are places where the DNA replication fork encounters lesions which it cannot go past. The first of these are UV-induced photodimers, cyclobutane dimers, which are formed primarily between two adjacent thymidine residues. And these photodimers really block the progression of the DNA replication fork and can lead to the breakage of these molecules by some kind of either torsional stress or, in fact, by enzymes which cleave the DNA. Another way in which these replication forks are stalled are by unusual DNA structures which form ahead of the DNA replication fork and which make palindromes and other structures which, again, are difficult for the DNA replication machinery to bypass. These happen in so-called triplet repeat sequences, such as CTG, 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 repeated many, many times, which is characteristic of a defect that is seen in Huntington's disease or in other DNA sequences which are characteristic of diseases such as Friedreich's ataxia. And in both of these cases, the stalling of the DNA replication fork creates an opportunity for chromosomes to break. There are lots of fragile sites in human chromosomes, which are sites where this kind of replication process is exaggerated. And so these are sites where, in fact, you see frequent chromosome breakage by looking, again, at the karyotype, as I showed you before. And in fact, one can reveal that these sites are particularly fragile by slowing down the normal DNA replication process using a chemical inhibitor, in this case called acetylcholine. And if one treats the cells with acetylcholine, what one sees here is that there are now many of these chromatid breaks which have occurred, very much like I showed you in the absence of the RAD51 protein. There are such breaks at a very low level in normal cells, but they are exaggerated tremendously by adding this inhibitor to DNA replication. So we know there are double-strand breaks that arise in cells, and they can be repaired in a variety of ways. There are two large categories of mechanisms by which these breaks can be repaired are called either homologous recombination mechanisms, in which the ends of the double-strand break have to find a template, a nearly identical DNA molecule that they can use as a matrix in order to patch up the broken chromosome. And there are a number of different ways in which I'm going to illustrate how this repair can take place. And then there are also other mechanisms which are called non-homologous end joining mechanisms, in which the ends of the DNA are pretty much just joined back together with little or no regard for DNA sequence identity. These joining events are inherently mutagenic because they often involve the loss of a few base pairs or more of DNA, but they are an efficient way of also repairing double-strand breaks. And then sometimes, as I showed you at the beginning, there are truncations, and many of those truncations involve simply adding new telomere sequences to the ends of double-strand breaks. That's a mechanism that I'm not going to talk about in this lecture.